scientists at Warwick HRI are breeding new types of brassica, such as broccoli, cabbage and oilseed rape, that are resistant to disease. In a moment, we'll be hearing from one of the researchers, Dr. Guy Barker. But first, here's his colleague, Dr. John Walsh.
Plant virus is a very important factor worldwide, affecting all crop types, but particularly vegetables. Not only do they reduce yield dramatically, they can have very severe effects on the quality of the produce. And in the UK, that's particularly important. In the supermarkets will often reject consignments of vegetables that have very minor symptoms. In this country, viruses are particularly severe because we're having these very mild winters, and also we have the Gulf Stream that protects the plants, and also the insects that transmit the viruses. So the insects are overwintering and spreading the viruses earlier. So it's more of a problem in this part of Western Europe and the UK than in parts of Central Europe, where they have much more severe winters. We work with a number of viruses, but the particular virus that's causing this syndrome is called turnip mosaic virus. You can see the terrible damage from this cut section of this cabbage plant, and the growers call this cigar burn, or they call this problem, this syndrome, cigar burn. You see these black necrotic spots that affecting the cabbage. So when the growers send a consignment of cabbage like this to the processors, you can imagine what happens. It's rejected. Which line are we looking at, Jude?
This one.
Okay, right. And what's happening? Turnip mosaic virus is transmitted by aphids. This particular aphid is called Mysis persicae. Aphids are more commonly known as green fly. So Mysis persicae is a very common aphid, and it's a very important virus vector. But it's particularly difficult to control because there are a number of insecticide-resistant strains around. Also, insecticides are very poor at controlling viruses like turnip mosaic virus because they're transmitted very rapidly by these aphids. The aphids don't even need to feed on the plant, they only need to probe, and they're able to transmit the virus. Also, they can pick the virus up from plants without feeding, again they only need to probe. So insecticides are particularly poor at controlling our virus. Here our research focuses on the use of natural plant resistance to control the viruses. So in the past, people have focused on these single R genes that will control the virus, but often they only control one strain of the virus. So in recent times, we've found some new sources of resistance where it appears to be broad spectrum, and so it appears to be controlling all isolates of the virus. So we've taken isolates from different parts of the world, China, America, Australia, etc., and we haven't been able to overcome this resistance with those isolates. So we're very optimistic this is going to give us durable, long-term control of the viruses.
Basically, I try and identify at a minute level the genes that are involved in regulating his trait of interest, which is resistance to the virus. So where he's been using genetics to identify quite a broad region of the genome which may be tied to this resistance, I then have to try and identify the few genes which are actually responsible for causing the resistance that he's detecting. What we do is we use markers that we've designed every few megabase across the genome. So basically we divide the genome up into fragments, and by using those markers we can then identify which fragment is initially responsible. We then take further markers and subdivide that fragment until we can get and keep doing that process until we can narrow down and then use our knowledge of the genome itself and the sequencing that we carry out within the laboratory to identify the actual gene and the variants of the gene which are responsible for the resistance that we detect.
You can see here examples of extreme resistance and extreme susceptibility. So with the extreme resistance, we've identified two genes that are involved with that, and we've now got candidates for those genes, and we're now in the process of actually verifying that those are the genes that are involved in controlling this extreme form of resistance. When we've been able to prove that our candidate genes are the actual genes involved in this resistance, we will produce molecular markers that we can then pass on to the breeding industry, and they can use those molecular markers to actually move this resistance from our plants here, these particular plants, into commercial cultivars of brassicas. And we're involved in negotiations at the moment with two major seed companies who are interested in this resistance and deploying it in some of their commercial crops.
It's a long process. It's not something that's going to happen overnight. We've been working on several of these traits now for years because we haven't been able to get the full sequence of the genome, which would allow us very rapidly to make those steps. It's a project that we're involved with and that the international community is fully behind. So led by Korea, who have a very big interest in something called Chinese cabbage, because they make kimchi, which is this pickled cabbage that they eat a lot of. So compared to our interest, which is brassica, broccolis and cabbages and kales, for example, in this country, internationally there's a much wider range of vegetables which this can be applied to.
This will never eradicate viruses in vegetables, but it's a first step with this particular virus, hopefully eradicating it from certain brassica types. And if from the research we can build on that and look at other virus species and identify similar means of controlling the virus, then hopefully there are hopes for these other viruses.
It's very exciting. I mean, if we look at the effect of virus on yields and oilseed rape, which isn't obvious, we can see that that can make a difference of 10% in the yield of the crop. And if you look at how we're trying to produce sustainable crops these days, this is a significant increase in yield and will make a big difference to the amount of oil, if we take oilseed rape, for example, that we could harvest from a field.
That's all from us for this week. If you'd like to comment on any of the stories we cover on Warwick iCast, we'd love to hear from you. Please visit us at warwick.ac.uk/go/iCast. Until next week, thanks for watching and goodbye.